So our next presentation is a trio, and I'm very, very proud to be part of that trio. This is Jiang, who does not need an introduction, and Jiangbing Zhou, who actually also, I think, after 4 years now, or 3 years, also doesn't need an introduction. This is an amazing team at Yale, who Jennifer and I have been lucky enough to be able to work with over the past couple of years as we advance this program, which is one of the most exciting, novel, cutting-edge programs that I've been lucky enough to be a part of. And what we want to share with you all today is where we are. We've been working on this diligently, and it's been really, really complicated. And as we're starting to see more and more success and really moving the needle on bringing us closer and closer to a possible future clinical trial, we wanted to share that data with you. So I'm here representing myself and Jennifer, but Yong and Jiangbing are going to talk to you about the technology and what we know in the animal model, and then I'm going to share with you how we've taken that from the mouse model and what we're doing to try to move that to humans. Yong, Jiangbing? You're going to start, yeah?
Thanks, Allyson, for the introduction, and good morning, everyone. For those who don't know me, I'm an engineer on the team. So my responsibility is to, without disclosure, so my responsibility is, should I go through this slide? Probably not. I think you...
You want me to do it? He knows the slide, but I'm happy to do it. So really, the goal of this project, and I think why this project got funded and got incredible support from the NIH, is because we really created this bench-side to bedside model to go from discovery and preclinical work at Yale to clinical trials at Yale and Rush. And we were able to kind of put all the pieces together in this development program, all through the support of our community who developed this entire ecosystem of bench-side to bedside. And because of that ability to be able to truly translate drugs to humans, the NIH believed in us and got us to this program, and funded this program. And so we've been able to really support that over time. So we'll start with the preclinical program, and then we'll move to the toxicology program, and then hopefully the future for clinical trials. Now you can go.
Okay, so as I just mentioned, I'm an engineer on the team. So my responsibility is to create a carrier to deliver our therapeutic payloads to the right location. So this case is the neuronal cells in the brain. So our payload is a genome editor called CRISPR, which discussed by the last presenter already. So CRISPR actually originated from bacteria. Is a hallmark of bacterial defense system. So believe it or not, bacteria, just like human beings, could be infected by virus. So through evolution, this bacteria derived a mechanism called CRISPR, which can cut the viral genome. So in that case, it can kill virus. So over a decade ago, our scientists in the field figured out a mechanism, how to hijack the mechanism for human use. So, so the mechanism actually, as shown here, has a two component system, including cars. It's basically a working machinery, so like a scissors here, for example, cutting the genome, virus genome. There's a guide RNA, so basically the code to tell the machinery to the right location. So, but for CRISPR, the major challenge is how to deliver. As we discussed yesterday and today, in the gene delivery field, there's mainly two vectors available. One is a virus, AAV. We talked about AAV a lot this morning already. So another is the nanoparticles. So so I will talk about how how I, as an engineer, to really solve this delivery issue. So this slide basically shows that how our scientists have been engineered CRISPR for human use. So in addition to the original formulation, which can use for genomic cutting to remove the targeted gene, so the machinery has been engineered for other use, for example, for gene correction or epigenome editing. Basically, we can repair whatever defect in human genes, like UBE3A. We can correct mutations, and we even can repair the deletion. So as I mentioned, delivery is a major issue, right? So there's two major delivery carriers in the field, including AAVs, and which is most often used at this moment. And another is the nanoparticles. So unfortunately, AAV may not be the best vehicle for deliver our genome editors. As I mentioned, right, this is a CRISPR derived from bacteria. And it is an active machinery which can potentially cut your genome or modify your genome. So in this case, you can imagine if we deliver CRISPR editors use AAV, so this editor will be expressed, bacteria-driven editor will be expressed in human cells for a long time, probably over a decade. So you can imagine, right, it could mess up our genome in addition to the immune reaction. So, other than virus, nanoparticles could be choice, particularly lipid nanoparticles. But unfortunately, lipid nanoparticles may not be best for neuronal use, particularly in this case, for Angelman syndrome, because it's very difficult to deliver LNPs or other nanoparticles to the brain. Through intravenous administration, particles need to cross blood vessel and get into the brain. But the vessel in the brain is very different from vessels in the other organs. They form a mechanism called blood-brain barrier, which have been mentioned by previous speakers already. So most particles cannot cross the BBB well. But even local administration or deliver through lumbar puncture, right, just like ASO delivery, the particles cannot be dispersed in the brain well. Could be simply because they are too large. Most nanoparticles probably is over 60 a couple hundred nanometer, but the free space in the brain tissue actually is very small, probably less than 30 nanometer. So to overcome this issue, we develop a new approach we call a step engineering approach. So, step, we present a group of chemicals. It's a small chemical, it's about 2 kilodalton, very small. So those chemicals are designed to, with two arms, the one arm, intact with therapeutic payloads, in this case, genome editors. So another arm, is designed to interact with specific cell type or organs. So in this case, for genome editors, we can just conjugate them together, and then these small molecules, you can believe a lot, they work so well with interact with therapeutic payload so well. You only need to put 10 to 20 those small chemicals with our genome editor payload. In this case. So these chemicals can bring this therapeutic payloads into cells and release them. So the beauty of this approach is that step engineered genome editors, the protein-based editors called RMP, are very small. It's about 10 nanometer. It's perfect for brain penetration in terms of size. So compared to others, right, nanoparticles, probably over 100 nanometer. So of course, I would like to mention that step here is a concept. So we actually have synthesized a large library of stable chemicals. And different chemicals may have different properties. For example, some chemicals like neuronal cells, you can, after injection, they preventively edit the neuronal cells. Some like astrocytes, and some probably like other organs. But of course, for Angelman syndrome, our target is the neuronal cells in the brain. So, so in this case, we choose a specific step molecule which can edit the neuronal cells very well. Just give example here. This is testing this step RMPs in patient-derived neurons. The 2D neuron grow on flask. Basically, what we found is that single treatment can edit the most, over 90% of our neuronal cells in the dish. So the efficiency is also very high in 3D neurons. So this is a cerebral organoid. Is a sphere culture in 3D fluid, 3D form in fluid. So basically, we found that one single treatment really edited most cells within cerebral organoids. So efficiency could be up to 90%. So over the past few years, we have been synthesize step RMPs and then work together with Yong Jiang's lab to characterize step RMP for Angelman treatment in preclinical studies. So Yong.
So when I relocated from Duke to Yale end of 2019, then I met Jiangbing roughly middle of the COVID 2021. So we kind of learned he has this technology. So my lab worked on a panel of disease. So kind of say, okay, maybe this is a great potential to picking disease, genetic disease, and to develop a treatment. As you probably know, the first choice is Angelman syndrome. That's my favorite, dear to my heart. So I don't think I need to go on to this slide too much detail. So the idea is this STEP technology, same as antisense oligo. You can inactivate antisense. The difference from antisense oligo is you need to repeat it multiple times, keep going. For STEP technology, it's a CRISPR editing. If it works, by design, it's only one time, permanent inactivated antisense. That's it. So that's the beauty. So just shows you the concept has been shown by ASO, ATF, and topoisomerase small molecule, and Cas9 in use AAV vector, and also Cas RNA inactivation. So it's basically, again, this is you are very familiar with, and the paternal chromosome antisense is very long. If you can inactivate it, transient or permanently, and then you can reactivate a gene from maternal chromosome. We use a deletion as a case, but presumably this technology should be applied to all other different genotypes. So this is a slide when Xiaona Lu was a postdoc at that time, and now she's a research scientist, shows me particularly on the right side of that particular Western blot, it's a protein analysis. When she showed me this plot, I immediately got quite excited and convinced. I told her, I said, we got a program. This is working beautifully. So just shows you that you can see there's no treatment, it's red, there's no very minimum protein with 3 sample treated, and from the mouse brain, it shows a beautiful reactivation, very clean. So on the same thing, we validated, she has done like almost more than 20 cohorts, several hundred mice, validated all the funding pretty efficiently. And one of the beauty for the STEP IMP versus the other delivered technology, AAV, we deliver the Cas9 protein, as Jiangbing alluded to. This protein goes into the in vivo, we do the intrathecal injection, which shows you here that green signal. You can see after day 1 injection, 1 hour, 3 hours, 6 hours, 12 hours. By 12 hours, by 24 hours, almost you cannot detect any Cas9 protein. As you probably all quite familiar with when talking about CRISPR editing, we want to fix the genetic mistake here is inactivated. You also want to make sure this editing not go to the other part of the genome, create another genetic condition. But with this technology, because so short transient presence in the cell, so to definitely minimize the potential off-target event, which is one of the major concerns for CRISPR editing. There's a lot of advantage for CRISPR editing. The major concern is the safety. So with this, and then we also demonstrate using the animal model we generated many, many years ago when I was at Dr. Bodas's lab, and as many people use this line too. Just shows you, this is a very busy slide, a lot of work, to get a few key points. One is we deliver the CRISPR editing at day 1 of the mice after birth, day 21, day 42. In this particular Lodora model assay, it's almost very, very similar regardless, day 1, day 21, and day 42. So that's the one point, key point. Second key point, which shows multiple Cas9 all working well for STEP technology, which shows you the STEP may be able to adapt to many different types of the Cas9 or other editor. This is Allyson shows you this slide yesterday, and it just quickly shows you with the content, which you clearly see this AS treated with CRISPR doing much, much better, all similar to the wild type. We have, Xiaona Lu have done a battery of a lot of behavior testing. Because of the time, I just showed you a few key ones. The second one we just show you this seizure susceptibility. Clearly, with this paradigm developed by a few part of UNC, you can see that, again, different day 1 and day 21 treatment, they significantly suppress the seizure threshold in Angelman mouse model. So with that, I'm going to turn to Allyson.
Great. So what you can see is, he called me and said, "You're never going to believe this. There's this guy at Yale, his name is Jiangbing Zhou, and he has this amazing technology, and I think it works for Angelman syndrome." And I thought he's going to show me a couple cell lines, and we were going to get excited and fund a grant to see if it worked. But instead, he showed me all of this animal data that Shauna had been doing, and what he showed was not only that it rescued every phenotype, but it rescued it at newborn, at juvenile, and at adult ages. And in some assays, adults did better than the newborns. So I was like, we have to support this. This is incredible. And so this is where we decided we're going to create a team, work together, and figure out a way to get this to humans. Because as you know, really the only thing I care about is getting out of a mouse and getting into a human. And that's where my strength is what can we do to move this out of an academic lab and actually get it into a clinical center. And so Jennifer, I recruited into this and said, we have to do this, and she fully agreed and was blown away, and she's not standing here, she's sitting there, but she's been a force with this program, and I'm gonna explain why, because it's actually really, really complicated. It's beautifully elegant, but also incredibly complicated, and she's been an unbelievable force on how we do that. So when we think about the translation, that next step to get from the Angelman mouse to an Angelman human, there's a lot of steps that we take. And I'm not going to walk you through every bit of it, but it's the safety assessments to understand, does it go where we want it to go, and is it safe? So the steps it takes to get through that is called the investigational approach to getting to a first-in-human clinical trial are IND-enabling studies. It's those studies required by the regulatory authorities and for us to feel comfortable that it's doing what we want it to do. And so those experiments are really assessing very critical information related to safety, related to toxicity, understanding if it's having any ill effects, but also is it targeting the goal? Is it targeting the antisense transcript? Is it knocking down the antisense transcript? And is it expressing the paternal UBE3A gene, ultimately making the paternal UBE3A protein? And is it affecting anything outside of that, right? That's the target, but what about off target? As Yang Wei mentioned, we don't want it to affect anything else other than the UBE3 antisense transcript that could result in a different genetic disorder. We're dealing with enough. We don't need a second genetic disorder. And so these studies are really critical in order to get there. And again, those are called IND-enabling studies. And a lot of you are like, well, what's taking so long? We've now heard about this for 2 years, and why aren't you in humans yet? Well, these studies are really complicated. They take a very long time, and they're very expensive. So we really take all these rigorous activities, we work in parallel, we really bring in experts in the field to help us and support us in getting this done. And some of the most expensive aspects and the most time-consuming parts of this program lie in this period, where we have to understand what we call the pharmacodynamics. So these are big words, but ultimately it's, does the step RNP knock it down and express the UBE3 protein? Where does it go in the brain? Is it only in the brainstem? Is it only in the spinal cord? Is it in the cortex, the hippocampus, the parts of the brain that we know control the behaviors of people living with Angelman syndrome? And so we want to make sure it gets throughout the brain into the critical brain areas that we believe are going to improve the symptoms of Angelman syndrome. How are we going to deliver it? So that's called the route of administration. So ROA, route of administration. If we deliver it into the spinal cord, can we go from below at the lumbar spine like ASOs are delivered, as Jim showed you? Is this behave like an AAV or like an ASO? If we go from below, can it get to the brain? If we go from the base of the skull, does it get to the brain? Do we need to go with a little baby catheter, a little surgery into the ventricle of the CSF compartment? And is that going to give us the best distribution to the brain? These are critical questions, because if it's a single treatment therapy, we need to get the best biodistribution in the safest possible way. And we need to be able to prove that, because this is an untested therapeutic modality. We have a lot of data on AAV gene therapy and how it gets to the brain and where it goes in the brain. We have a lot of data on ASOs and how it gets to the brain and where it goes in the brain. But we have no data on this very new CRISPR technology, because it's not being carried by an ASO or by an AAV or by a lenti. It's being carried by this novel STEP component. And so we have to really answer those questions. And then, is it safe? At what dose level that we're testing this, a low dose, a mid dose, a high dose, how does that translate from mouse to monkey? How does that translate from monkey to human? And we have to understand, is it safe at the doses that give us the best brain distribution and that allow for the best UBE3A paternal gene expression? And so these are very critical questions that we had to ask. And so really what I want to show you is what you probably are going to register and remember, is that we had to then put it in monkeys, right? And so this is really important, because a mouse is not a monkey, and a monkey is not a human, but a monkey is closer to a human. And so we want to know, what is the best way to get it there? Is it safe? And how do we get to the largest portion of the brain? So we looked at a comparative study of monkeys using IT, lower spine, like an ASO, ICM, back of the skull, like an AAV gene therapy, or ICV, into the ventricle through a small little catheter that goes into the ventricle in the brain. And if you look at this at a half an hour on the left of each one, at 24 hours in the middle, and at 72 hours on the right, what you can see is pretty obvious. You can see you get much more of the compound to the brain with ICV delivery than you do with IT or ICM. And so we really can see this what we call biodistribution to the brain, and the route of administration is going to be best if we can deliver it that way. And doing that, because it's hopefully just a single treatment therapy, is not a hard thing to do. If we had to do that multiple times with like an ASO, that would be very complicated, because it is a small surgery. It's neurosurgery to put a small catheter right through the skull and into that compartment. But a single treatment therapy, that's possible, and many gene therapies are actually using that approach, because it's actually a little bit easier. A lot of neurosurgeons know how to do that, to train people to do that. Even though the ICM is a little bit less invasive, ICV is possible because a lot of neurosurgeons know how to do it. But it is surgery. And so we needed to answer that question, and these experiments really allowed us to be able to do that and to be able to show that very nice distribution in these monkeys over time. And so that has taken almost 2 years to get to this point, and what we can see is that it's tolerated. These doses, the monkeys are surviving, and we are able to see that it goes throughout the brain. By 72 hours, we really see, even at 24 and a half an hour, we're seeing a ton of it in the brain. And so when we put that into a graph, what you can see is that the entire brain has the best distribution of the radioisotope that's being captured by PET CT with the ICV delivery. And that is the first step to now the next stages of the program. With that, there's the manufacturing of this product, and I need to have a shout out to Jennifer and Conrad and Derek. I am only presenting this in the most simplistic way, because I remove myself from all manufacturing conversations because I find them maddening and frustrating, and they're too complicated for me to actually understand. So I'm being an imposter at the moment, but ultimately for manufacturing, it's really complicated. We have to make the final drug using what we call the drug product in a way that not only is safe in animals, but has to be the cleanest possible way, the cleanest possible substance for people. And that's very complicated. It's expensive, it's complicated, and it takes a long time. And for this program, we have a guide, we have a Cas protein, and we have a step molecule. That means 3 different manufacturing processes need to happen, and the quality of that and the team that's working on that has been unbelievably incredible in order to get that to what we call a good manufacturing process for people. And so that takes a lot of time and a lot of money as well because of those complicated 3 components, and there's not a track record like ASOs or gene therapy on how you manufacture this and complex it together. So if you put all of that together, which hopefully you may think is very, very complicated, but I've tried to simplify it in a way in which you can see all of these pieces. You start here and you go, you go, you go, you go. This is a drug development program. It's very, very complicated in order to get to the goal, which is the only thing I actually care about, which is getting to first in human. We have to be able to show that it works as well in a human as it does in a mouse, because the mouse only tells us so much. But it takes a ton of work for the safety piece, for the manufacturing piece, for the understanding of age and distribution piece. And all of that together is what drives drug development. And this is a program that is being driven by so many incredible people that are all contributing to all of these pieces. And I'm just representing this in one simple picture. And so, ultimately, these studies that need to bring human candidate to human application take a tremendous amount of coordination, a tremendous amount of time and money, very careful coordination between the real critical teams, preclinical, non-clinical manufacturing, regulatory, clinical. All of this takes a tremendous amount of collaboration, and every step of this, in drug development in general, there's unexpected findings. Sometimes you're like, well, the dose that worked in mouse didn't work in monkey. Well, the dose that worked in mouse worked too well in monkey. We have to lower the dose. So you have to do all of these studies to make sure you're really stepping up in a way that is going to provide the most information to ensure the rigor and safety for humans is of utmost importance, and that can take time. So ultimately, with all of the work that was done here, we were able to leverage a grant from the NIH in order to allow us to leap from mouse to human, and that is the drug development ecosystem. And ultimately, as we have done this discovery and preclinical work and we're moving toward the acceleration of this clinical work, it's very exciting to see this program moving forward, day after day and month after month, and it's super exciting to share that with you. So in conclusion, the program shows incredible support through infrastructure and collaboration, bringing many teams together with various expertise to allow for efficiencies that really can't be achieved in any other way. If you don't get out of your silo, you get in your own way. And so everyone on this team has gotten out of their silo, leveraged the expertise of others, and allowed them to work together to really advance this program. And the fact that Angelman syndrome is serving as an example for so many other rare neurogenetic disorders has allowed us to create this platform for future personalized medicine, not only for Angelman syndrome, not only for single mutations that might benefit from one different type of therapy or another, but utilizing this platform, but for other neurodevelopmental disorders as well. And so it's wonderful, and it feels great for Angelman to be that platform to help so many other disorders. And as you know, at FAST, we work really hard to ensure that we want to be the low-hanging fruit. We want to be the example, but we must ensure that the example we show helps others, that we cannot advance the field if we don't do that. So we are really leveraging our preclinical and our IND-enabling work and our clinical capacities throughout this network to be able to help many other diseases. So seriously, we could not do this without the support of the NIH. We got an over $40 million grant to support this work. All of this work started in a lab at Yale. So to these guys, I'm only presenting the part that matters to me, which is getting to humans, but we couldn't do it without the unbelievable technology that they've developed, and the commitment to the science that they have had has been incredible. And we can't wait to bring Rush into the fold, because the clinical trial will, of course, be supported by Yong at Yale and by Liz at Rush. And so hopefully the next steps is when we can get to that clinical piece, which we're just not quite there yet. So 1,000 people to thank. I'm not going to thank the Yale team. They should thank the Yale team. But what I can say is that the collaboration of all of these people has been so unbelievably incredible. There is such a commitment to this program by the team at Yale and Couragine and Courageous and FAST and AS²Bio. Jennifer has just been a force to be reckoned with, and really been leveraging the regulatory expertise to get something so new and so unique over the finish line, and really bring that to humans in a way that's safe, that's going to be agreeable and applicable for human application. And so it's been a real honor to work with this team, and I'm proud to represent them up here. So with that, Thank you to all of these people that have been such a huge effort in this program. Thank you.